Hey guys, in this video, I'm going to be going through the light reactions step by step. And just to start off, I want to reiterate that the light reactions happen inside of the thylakoid. So down here in this region is actually the inner thylakoid, or inside of the thylakoid. And up here in this region is actually outside of the thylakoid, or in that stroma, which is that kind of gel-like fluid the thylakoids float around in. So we kind of have the situation where we have inside of the thylakoid. This is the membrane that separates the thylakoid from the stroma, just like a cell membrane separates inside of the cell from outside, except for this time we're inside of a chloroplast. And thylakoids do have a phospholipid bilayer with proteins and photosystems, and they're going to help us make the products of the light reactions, which I'll get into in a second. So here we have the stroma, which is kind of like outside of the thylakoid. So I'm going to go through this step by step. And the first step of the light reactions is that light is going to strike our leaf, and it's going to hit our chloroplast, and it's going to hit our thylakoid, and it's going to actually do two things. The first thing is that it's going to split our water molecule. So it's actually going to split our water into three separate things. It's going to split and it's going to release some oxygen gas. And that oxygen gas is actually going to travel right through the center of the bilayer because it is a non-polar molecule. It can just diffuse right through. So it kind of bounces out of the cell. It is not used in this process at all. It is just waste. But what is useful is this water, in addition to being split into oxygen, is going to lose electrons, which means that it is oxidized. And it is losing electrons, and also it loses some H+. And actually, the inner thylakoid space, because water is kind of constantly being split, is going to have a lot of H+, and it's actually going to get H+ from the stroma as well, but we're going to get there in just a second. So far, water has been oxidized. We've lost an electron. We've lost some H+. Oxygen gas has been lost out of the cell, no longer used. So the next thing that's going to happen is that light is going to strike the electron, which is in photosystem 2. And photosystem 2 actually contains the light-absorbing pigment chlorophyll. So it's going to absorb light, and it's going to use that light it's absorbed to excite this electron, which is kind of like charging it up. So I think electrons can be used as energy if they have, if they're excited or kind of charged. So light is going to strike photosystem 2, and then that light that's absorbed is going to excite or charge our electron. So I always kind of do an arrow pointing up to show this electron is in a higher state of energy, or it has high potential energy. And I know in class I use the analogy, it's sort of like when you have your phone and you charge it up, it's going to be kind of at 100% battery. So you can kind of imagine this electron up here has been charged. So it's sort of at 100% battery, which means it is going to be very useful to us. So I am going to show that light is definitely coming in and striking the pigments in the photosystem 2. The only color that's really not going to be used is green, and that's why the photosystem is going to reflect green, but violets and reds are going to be very useful, and they are going to be used to charge our electron into a higher state of energy. So I'm just going to write all of those steps out. So we've got the light is split and oxidized water, which we've kind of lost this electron and it's gone to the photosystem. The H+ stays in the inner thylakoid. The oxygen gas diffuses through the membrane and out of the cell, out of the plant. And then light's going to strike photosystem 2 and send this electron to a higher state of energy. So high potential energy means we have the ability to do work. And that's what we're about to see. So we're going to move on to the next step. So in the next step, we're actually going to use our high energy electron, which I've kind of changed, and I put it in this little green ball to kind of help us visualize where it's going to go. So it's already been this electron has been charged up, so here we can actually use it. So I went ahead and drew in all the other H pluses, so you can see that down here in the thylakoid space, there is a high concentration of H plus, and up here in the stroma, not so many. So we're going to have a low concentration of H plus. And remember, drawing brackets around something like that just means concentration. So low concentration of H plus up here, high down here. So what's going to happen is this electron, we're actually going to enter what's called the electron transport chain. And the electron is going to move into the chain. And when it does that, this protein right here is actually going to use energy from our fully charged electron, and it's going to use it to take a H plus and force it into the thylakoid, or this inner thylakoid here. So it's going to take it and force it across the membrane. But we know from the last unit with the cell that whenever you force something from low concentration to high, that is called active transport, and that requires energy. Now in this case, it's not going to use ATP, but it is going to suck some of the charge or some of that energy away from the electron. So as the electron moves into E here, it's going to take an H plus and it's going to put it into the thylakoid space, or the inner thylakoid. So it's gone across, and in that case, we've actually kind of lost some energy in our electron. So that this electron here is sort of now at like 75% battery. So it was at 100, it's kind of used some of its energy to move that H plus across, and now it's going to move to the next protein in the chain, and it's going to take another proton and allow it to pass through into the inner thylakoid, and it's going from low to high again, and in this case we're going to use more energy from the electron. So if it was at 75, maybe now it's at 50%, and it's going to move to the next protein in the chain, which is going to take another H plus and force it through from low to high using energy, and now this electron is pretty much spent and it is at a low state of energy again here in photosystem 1. So the whole point of that electron transport chain is really using the energy from the electron, which got its energy from the sunlight here, and it used that energy to actively transport hydrogen ions, or H pluses, from the stroma into the thylakoid space. So what we have now is our electron that is not really charged anymore. It's kind of at low battery or dead battery. And what we do with our phones is when they're at low battery, we charge them up. But first, I am going to write out the steps to that electron transport chain in words before I move on. So here I've just written out what I just showed you in words. So the charged electron enters the ETC. The ETC uses that energy from the electron to actively transport H plus from low in the stroma to high in the thylakoid. So if you want to write that down or read it, I would go ahead and pause the video because I am going to erase it to move on to the third step. So step 3 is we're going to take this kind of uncharged electron and we are going to charge it back up using sunlight yet again. So sunlight is going to come in and strike the pigments in photosystem 1, and it is going to excite our electron once more. So it's kind of going from that dead battery to a fully charged battery. So now that our electron is charged up again, instead of being used to pump protons into the thylakoid space, it's actually going to be accepted and picked up by our electron carrier. So in this case, the electron carrier is a molecule called NADP+, which will be reduced because it's gaining this electron right here. It'll be reduced into NADPH, which is the first major product of the light reactions we've gotten so far. So NADP plus has been reduced since it has gained an electron, and NADPH is going to carry that electron to the carbon dioxide to help make a sugar. So that is actually step 3. So that step is a little bit shorter, so it's just exciting that electron in photosystem 1 using our chlorophyll pigments, and it's going to reduce NADP plus into NADPH, which leads me to the 4th and final step of the light reactions. You've noticed we haven't used ATP synthase yet, so what was the whole point of pumping all of these protons into the inner thylakoid? Well, we can actually use those protons to create ATP. So what happens is they travel from passively from high to low through ATP synthase, and it actually, as the H plus passes through ATP synthase, it creates ATP, which is the final product of the light reactions. So H plus is going to travel. It is facilitated diffusion since ATP synthase is an enzyme. It's an anabolic enzyme because it is actually going to build an ATP, and that is a high energy molecule. So it's literally as simple as our H pluses are going to travel through ATP synthase, and in the process, ATP is created, and we're actually going to keep that ATP. So to kind of summarize and tie it all together, just know that this electron that winds up at NADPH, it does come from water. Water is split, and that electron kind of makes its way into NADPH as it goes from photosystem 2 through the ETC and then into photosystem 1. And then the ETC is very useful because as the electron goes through it, it helps create this high concentration of H plus in the thylakoid and very low in the stroma. And that concentration gradient, or that high to low balance we have, is used to power ATP synthase and creates ATP. NADPH and ATP are the 2 products of the light reactions. But you're probably wondering, or maybe you aren't, where's the sugar? Because I thought photosynthesis was all about making sugars. Well, in the final stage of photosynthesis, it's called the dark reactions. And that is where we're going to take this NADPH and this ATP and add all of that energy to beef up our carbon dioxide and make a glucose. So the dark reactions actually occur in the stroma, which is that white gel inside of the chloroplast. And through a series of chemical reactions, which I'm not going to get into this year, that would be if you took AP biology, I would go into the details of the dark reactions. But what you need to know is that through a series of reactions, we add 18 ATP and 12 NADPHs, which, as you can recall, these guys were the products of the light reaction. So they were made in the thylakoid, or light reactions, and they are going to be added to the 6 carbon dioxides. And because this is an anabolic reaction, all of that energy is getting stored into the CO2, and NADPH is going to give all of those electrons that it got from the water, and it'll actually reduce carbon dioxide. So CO2 is actually reduced here. So think energy from ATP goes in, electrons from NADPH goes in, and NADPH is going to give its electrons or lose its electrons as it gives them to carbon dioxide, and it will actually become NADP plus again, which will go back to the thylakoids or the light reactions to grab some more electrons and bring back to the dark reactions to keep reducing our carbon dioxide. And that, in the end of the day, is going to give us our 6 carbon sugar, which, where did the 6 carbons come from? They came from our 6 CO2, so 6 1-carbon dioxides, to a single 6-carbon glucose, and it's C6H12, O6. And you can see we've got carbon and oxygen in carbon dioxide, but where are all these hydrogens coming from? Well, they're coming from the 12 NADPHs. So as the electrons are added into this carbon dioxide, we're actually adding in that H, right? So that H didn't just disappear. When NADPH becomes NADP+, it's lost electrons and those hydrogens, which have been used to make our final product of this entire process, which is our lovely glucose molecule. And as you can see, energy is going into, right? So think about these arrows. Energy is going from ATP made in the light reactions into the CO2. All those electrons and H pluses are in the H's are going into our CO2 as well. So all that energy, all those electrons are going in and being stored into this high energy glucose molecule, which the plant cell can then either store it, turn it into polysaccharides like starch. So think glucose is a monosaccharide. We can string a bunch of these together to make starch, and can also be used and broken down in the mitochondria to make ATP to power all kinds of processes, which is actually cellular respiration, which we'll get into later on. So in a nutshell, this video has covered the entirety of photosynthesis, from splitting water, using sunlight, using carbon dioxide, using the light reactions, and ATP, NADPH to make a glucose molecule. So it's a lot of work just to get a single glucose, but here we are, and this glucose is going to be used not just to power the plant, but also to power me and you, because this is where our food originates from. So thanks, plants, and our oxygen we breathe too. So that in a nutshell is our photosynthesis.